Hello students, welcome to this digitized Form 4 biology course. In Form 3 we covered the topics Classification 2, Ecology, Reproduction and Growth and Development. In this course we will cover 4 topics. These include genetics, evolution, response and coordination, and support and movement. Genetics. Introduction. Genetics is the study of heredity and variation. Heredity is the transmission of characteristics from parents to offspring during reproduction. Variations are observable differences in organisms of the same species. For example, heights in a family, some may be about continuous and discontinuous variation, the first law of inheritance and describe Mendel's work, sex determination and linkage, mutations, and application of genetics. Concepts of genetics. Variations within plant and animal species. There are 2 types of variation, discontinuous variation and continuous variation. Discontinuous variation. This is the possession of observable differences where individuals form definite distinct groups without intermediate forms. For example, an individual is either male or female, tongue rolling, hair in the nose or ears, and fingerprints. Discontinuous variation is controlled by 1 or 2 major genes whose physical expression is not influenced by environment conditions. Play the video clip to view rollers and non-rollers. Continuous variation. Continuous variation, on the other hand, is characterized by possession of observation differences where individuals exhibit a wide range of the same characteristic from the extreme end to the other with many intermediate forms. For example, height. It's controlled by many genes whose expression is affected by the interaction with the environment. The photograph shows individuals of the same age but different height. Causes of variations. Variations are caused by gene formation, fertilization, and mutations, sudden changes occurring in the gene or chromosomes. Play the animation to see formation of chiasmata and crossing over in meiosis. Chromosomes. We learned earlier that chromosomes are thread-like structures found within the nucleus which carry the genetic materials. Each chromosome is made up of two parallel strands called chromatids connected by a structure called centromere. All chromosomes occur in pairs looking alike and each has a characteristic length, though they contain different genetic composition. The member of each pair is known as homologous chromosome. The illustration shows the structure of a chromosome. Each species of animals or plants contain a definite constant number of chromosomes. For example, in man, a sperm has 23, while an egg has 23, making the zygote to have 46 chromosomes. Play the animation to view formation of a zygote. Chromosomes exhibit certain characteristic behavior during the stages of cell division. On each chromosome, there are several genes which are shorter sections of DNA, and each gene determines a particular characteristic of the cell in an offspring. The genes occupy definite positions on the chromosomes called gene loci. A gene is a portion of a DNA that codes for a protein. DNA is a complex molecule composed of building blocks called nucleotides. The illustration shows a DNA molecule. Each nucleotide is comprised of one of the four bases, namely cytosine, guanine, adenine, and thymine, deoxyribose sugar, and a phosphate molecule. The illustration shows the structure of a nucleotide. First law of inheritance. Mendel's experiments. Gregor Mendel, an Austrian monk, carried out experiments to investigate inheritance of certain contrasting characteristics of the garden pea plants. These characteristics of the garden pea plant included height of the stem, dwarf or tall, texture of the seed coat, wrinkled or smooth, color of the seed coat, yellow or green, and color of the pods, green or yellow. The photographs shows various characteristics of garden pea plant. If pure breeding yellow seed pea plant is crossed with pure breeding green seed pea plant, all the offsprings produced yellow colored seeds. Play the animation to view the cross. If the F1 yellow seed peas are crossed, the F2 generation produces green and yellow in the ratio of 3:1. This shows that the yellow color characteristic is dominant over the green color. Play the animation to view the cross. We can conclude that in the F1 generation, there is only one color of seeds, that is yellow, expressed although one of the parents had green seeds. Such a character which is not expressed physically, although it is present in the individual, is called a recessive character. This can be represented in a Mendelian cross as shown on the figure. Mendelian first law of inheritance. The characteristics of a diploid organism are controlled by alleles occurring in pairs. Of such allele, only one can be carried in a single gamete. Punnett square. An alternative way of showing monohybrid cross is by use of Punnett square or checkerboard. In a Punnett square, the parental genotypes are written outside a graphical grid. The gametes of one parent are written along the top of the grid, while the gametes of the other parent are written down the side of the grid. The products of the various fusion of gametes from the two parents are written in the appropriate boxes in the grid. Their relative ratios can be determined from the grid. Play the animation to see monohybrid cross by use of a Punnett square. Terminologies in monohybrid inheritance. Phenotype. This is the physical characteristic of an organism resulting from the influence of the genes and environment. For example, plant can be tall or dwarf. Genotype. This is the genetic constitution of an organism. A phenotype can be produced by more than one genotype. Examples of different genotypes are shown on the figures. Pure breed. These are individual plants which, when self-fertilized, produce offsprings which are phenotypically and genotypically similar. Allele. One of the gene pairs that determine a character. Allele always occurs in pairs. For example, the character for stem height tall can be produced by either allelic pair capital T or capital T small t. The illustration shows examples of allelic pairs. Dominant allele. This is the allele that influences the characteristics to develop in an individual over another allele though present. For example, allele capital T for tall plant will be expressed as tall plants whether in homozygous state capital T or heterozygous state capital T small t, irrespective of presence of allele for dwarfness small t. Recessive allele. This is the allele that cannot influence a characteristic to develop in an individual when present with dominant allele. It is usually represented by a small letter version of the letter representing dominant allele. A cross between dominant and recessive genes is shown on the figure. Diploid. This is the number of chromosomes contained in the somatic body cells denoted by 2n. Haploid. This is the number of chromosomes contained in the sex cells or gametes which have the diploid number and is denoted by n. The illustration shows diploid and haploid cells. Homozygosity. This is where the two alleles in a genotype are similar. For example, double capital T or double small t. Heterozygosity. This is when the two alleles in a genotype are different. For example, capital T and small t. First filial generation F1. These are the resulting offsprings or progeny of the initial parent in a genetic cross. Second filial generation F2. These are the offspring resulting from the cross of individual of the first filial generation. The first and second filial generations are shown on the following illustrations. Complete and incomplete dominance. Characteristics in an organism are controlled by genes inherited from both parents. An allele is the alternative form of a gene located at a particular site on a chromosome. The illustration shows an example of an allele. Complete dominance. A characteristic in an individual is controlled by the two alleles. If one of the alleles suppresses the other such that its effects are not observed in the presence of the other allele, then this is complete dominance. This allele is said to be dominant, while the one that cannot express itself in the presence of the dominant allele is said to be recessive. An example of complete dominance is shown on the illustration. Play the animation to see an example of complete dominance in maize plants. Incomplete dominance. There are situations where there might be no complete dominance and each of the alleles expresses itself in an individual. An example of incomplete dominance is shown in the following cross. Play the animation to see an example of incomplete dominance. In this example, neither of the alleles was dominant over the other. They show blending. This is incomplete dominance. Inheritance of the ABO blood groups and rhesus factor. Inheritance of the ABO blood groups. The major blood group system in humans is the ABO blood group. Using this system, individuals can be of blood group A, B, AB, or O. The blood groups are inherited. Inheritance of blood groups is controlled by 3 genes, A, B, and O. These genes occupy the same locus on the chromosome. Genes A and B are equally dominant or codominant, while gene O is recessive to both A and B. Chromosomes occur in pairs. Each individual can only have 2 of the same genes. This is illustrated on the diagram below. The presence of a single dominant gene results in the production of a protein called antigen on the membrane of the red blood cell. Gene A leads to production of antigen A, while gene B leads to antigen B. Gene O does not result in the production of any antigen on the red blood cells. The antigen on the red blood cells determines the individual's blood group. The presence of a single dominant allele also results in the blood group producing antibodies which float in the blood plasma. Gene A results in the blood-producing antibody B, while gene B results in the blood-producing antibody A. For example, an individual with genotype AO would give rise to antigen A on the red blood cells and antibody B in the plasma. An example is shown on the illustration. The following are therefore the various genotypes in the ABO blood system with their corresponding antigens, blood groups, and antibodies in the plasma. The illustration shows the various genotypes in the ABO blood system. The rhesus factor. This is a protein or antigens found on the surface membranes of red blood cells. It occurs in 85% of people. 15% do not have it. Those who have are described as rhesus positive, indicated by adding the positive, and those without are indicated by adding negative symbol to the letter for the blood group. The table shows various blood groups with or without rhesus factor. The presence of the rhesus antigen on the red blood cells is genetically determined. If the male parent is AB positive and the female is OO positive, then all children of the two will have the rhesus factor positive. This cross is demonstrated on the illustration below. If one of the parents is rhesus negative and the other parent is rhesus positive, all the children will be rhesus positive due to the rhesus gene present in the blood of the other parent. For instance, a male parent AB positive and a female parent AB negative will have all children being rhesus positive. The illustration shows the cross between the two parents. Sex determination. As we learned earlier, there are 46 chromosomes in every somatic cell in humans. The genes that determine whether a child conceived becomes a male or female are located on specific pair of chromosomes known as sex chromosomes. The sex chromosomes are given the symbols X and Y due to their shape. The illustrations show X and Y chromosomes. In females, the sex chromosomes are identical. They are of X type. Play to see the sex chromosomes in females. In males, the pair is XY, which are non-identical. Play to see the sex chromosomes in males. The sex of a child is a matter of chance. It depends on whether the spermatozoon that fertilizes the ovum carries the X or the Y chromosome. The illustrations show sperms carrying different types of sex chromosomes. During fertilization, there are 4 possible genotypic combinations. Half of the combinations show that the child will be a girl, while the other half shows that the child will be a boy. The illustration shows sex determination in humans. Therefore, the probability that a boy or girl is produced is a half. The illustration shows the probability of a girl or a boy being produced after fertilization. Linkage. As earlier discussed, pair of homologous chromosomes always carry genes for the same characteristic at the same place on the chromosome. The illustration shows gene loci in a pair of chromosomes. Apart from determining sex of a child, the sex chromosomes have other genes as well. These genes are said to be sex-linked. All linked genes constitute a linkage group. Linked genes are inherited together. They do not segregate during meiosis. The illustration shows chromosomes with linked genes. sex-linked genes. These are genes located on the photographs show some characteristics controlled by sex-linked genes. Linkage. As earlier, sex-linked genes. These are genes located on the sex chromosomes and are transmitted together with those that determine sex. Most sex-linked genes are carried on the X chromosome. The Y chromosome carries very few genes. In humans, there are few genes located on the Y chromosome which control characteristics that are exclusively male. These include premature baldness and tuft of hair in the ear pinna and in the nose. Some of the characteristics controlled by genes on the X chromosome include color blindness and hemophilia. The two can appear either in males or females. The photographs show some characteristics controlled by sex-linked genes. The photographs show some characteristics controlled by sex-linked genes. Mutation. It refers to spontaneous linkage. As earlier discussed, apart from determining sex of a child, the sex chromosomes have other genes as well. These genes are said to be sex-linked genes. Color blindness. A colorblind person is unable to distinguish between red and green colors. This condition is referred to as red-green color blindness. The trait is linked to the X chromosome. The gene that determines normal color vision is dominant over that for color blindness. This is therefore illustrated using capital letter C for normal color vision and small letter c for color blindness. Since the genes are linked to X chromosomes, its alleles are represented as X capital C and X small c. Play the animation to see the genotypes for color blindness and normal vision. A cross between a colorblind man and a woman homozygous for normal color results in daughters being carriers but with normal color vision and sons having normal color vision. The daughters are described as carriers since they are heterozygous. The X chromosome inherited from their father had color blindness gene linked on it. They will, however, have a normal color vision because the gene for normal color vision inherited from their mother is dominant over the gene for color blindness. The illustration shows the cross colorblind man and a woman homozygous for normal color. Hemophilia. This is a sex-linked trait where the blood of the individual takes an abnormally long time to clot after injury. The result of this is usually prolonged bleeding, hence the term bleeder's disease. Hemophilia is caused by a recessive gene on the X chromosome. This condition is normally illustrated using capital letter H for normal blood clotting condition and small letter h for hemophilia. Since the genes are linked to X chromosome, its alleles are represented as X capital H and X small h. Play the animation to view the genotype and phenotype of a hemophilic and a person with normal blood clotting. In a cross between a man with normal blood and a woman who is a carrier for hemophilia gene but with normal blood clotting, some of the daughters will have normal blood clotting and others will be carriers. For the sons, some will have normal blood while others will be hemophiliac. The illustration shows a cross between normal man and a carrier woman. Mutation. It refers to spontaneous change in the individual genetic makeup. It may lead to individuals with some unusual characteristics which may be beneficial, neutral, or harmful. Mutations are normally due to recessive genes. Mutations can occur naturally. They can also be induced by certain factors called mutagens. Examples of mutagens include radiations, mustard gas, colchicine, and hydrocarbons. Types of mutations. There are two types of mutations: chromosomal mutations and gene mutations. Chromosomal mutations. Chromosomal mutation may be the result of change in the number or structure of chromosomes. The 5 types of chromosome mutation are deletion, duplication, inversion, translocation, and non-disjunction. Deletion occurs when some section of chromatide breaks off and fails to reconnect to any of the chromatides. Deletion involves loss of genes. Play the animation to see how deletion occurs. Duplication. In duplication, a section of chromatide replicates and adds an extra length to itself. This leads to repetition of set of genes. Play the animation to see how duplication occurs. Inversion. Inversion occurs when a chromatide breaks at 2 points, then rotates through 180 degrees, then rejoins in an inverted manner. The nucleotide in the sequence is reversed. Play the animation to see how inversion occurs. Translocation occurs when a section of a chromatide breaks off and becomes attached to another chromatide, but on the non-homologous pair. It involves movement of genes from one non-homologous chromatide to another. Play the animation to see how translocation occurs. Non-disjunction. Non-disjunction leads to addition or loss of one or more whole chromosomes. During anaphase 1, the 2 homologous chromosomes may fail to segregate and so move on to the same gamete cell. If the sister chromatide failed to segregate at anaphase 2, the 2 chromatides move to the same gamete. Play the animation to see how non-disjunction occurs. Disorders due to non-disjunction. Non-disjunction may lead to various disorders referred to as syndromes. Such syndromes include Down syndrome, Klinefelter's syndromes, and Turner's syndrome. The photograph shows examples of disorders that result from non-disjunction. Down's syndrome. Klinefelter's syndrome. Individuals with Klinefelter's syndrome have an extra sex chromosomes, hence they have a total of 47 chromosomes in their cells. Symptoms of Klinefelter's syndrome in males include underdeveloped testes, breast development, reduced facial hair, and taller than average with signs of obesity. Persons with XXX constitution appear relatively normal in most characteristics. Symptoms of Klinefelter's syndrome in females include underdeveloped ovaries, characteristic facial features, constriction of the aorta, web of skin on the neck, and poor breast development. The photograph shows a person with Klinefelter's syndrome. Turner's syndrome results when individuals have one of the sex chromosomes missing due to non-dysfunction. The individual, therefore, has 45 chromosomes instead of 46. YO zygotes do not develop due to absence of many vital genes. XO are females with underdeveloped female characteristics, some of which include rudimentary ovaries and testes, small uterus, broad chest and widely spaced nipples, low hairline, low set ears, increased weight, obesity, small fingernails, no breast development, short in stature, and attention deficit disorder, that is, problems with concentration and attention. Gene mutations. Gene mutation involves change in the structure of a gene. It arises as a result of a change in the chemical nature of the gene. The change may also involve alteration in the DNA molecule. This alters the sequence of amino acids during protein synthesis. The result is an unintended protein molecule which may bring confusion or may be lethal. Gene mutation includes insertion, deletion, substitution, or inversion. Insertion. It refers to addition of a base into an existing DNA strand. This alters the amino acid alignment. Play the animation to see how insertion occurs. Deletion. It refers to removal of a gene portion. For example, if a gene is deleted from its position, the base sequence becomes altered. Play the animation to see how deletion occurs. Substitution. It refers to replacement of a portion of the gene with a new portion. For example, if A is replaced with G on a DNA strand, the base sequence is altered. Play the animation to see how substitution occurs. Inversion. A portion of DNA rotates 180 degrees. The inverted portion rotates, resulting in alteration of base sequence. Play the animation to see how inversion occurs. Disorders due to gene mutations. In the earlier lesson, you learned that there are many types of gene mutations. These mutations lead to disorders in the human body which are hereditary. The common hereditary disorders include albinism, sickle cell anemia, hemophilia, and color blindness. In this lesson, we shall learn more on those disorders. Albinism. Albinism is a condition where the synthesis of the pigment melanin fails and is characterized by a light skin, white hair, and pink eyes. The pigment melanin is a derivative of the amino acid phenylalanine and tyrosine. The pigment is synthesized through a series of enzyme-controlled reactions. Each of these reactions is controlled by a specific gene. In albinism, the gene undergoes substitution and a recessive allele designated by a letter small a. Under homozygous condition, double small a, the enzyme tyrosinase is not synthesized and consequently melanin is not formed. Tyrosinase is necessary to act on tyrosine, which is required in melanin formation. The photograph shows a person with albinism. Sickle cell anemia. This is a condition where the red blood cells contain abnormal hemoglobin, making the red blood cell to have a sickle shape instead of the normal disc shape. The illustrations show sickle-shaped red blood cells and normal red blood cells. The photograph shows sickle-shaped red blood cells. Hemophilia. This is a sex-linked trait where the blood of the individual takes an abnormally long time to clot after injury. The result of this is usually prolonged bleeding, hence the term bleeder's disease. Hemophilia is caused by a recessive gene on the X chromosome. This condition is normally illustrated using capital letter H for normal blood clotting condition and small letter h for hemophilia. Since the genes are linked to X chromosome, its alleles are represented as X capital H and X small h. Practical applications of genetics. Knowledge in genetics has been applied in a variety of scientific fields such as blood transfusion, plant-animal breeding, genetic counseling, and genetic engineering. Blood transfusion. This is a process of transferring blood from one person, who is the donor, to another, who is the recipient, in order to save the life of the patient. The video clip below shows a blood donation process. Click on the play button to view the video.
Yes, found. For 2 stages. Yeah.
Was rollen we nou meer.
There you go.
Play the video clip below to view the blood transfusion process. For the transfusion to be successful, the donor's blood group should be compatible with that of the recipient. To ensure compatibility of donor and recipient blood group, blood typing is done prior to transfusion. During blood typing, a qualified person tests for both ABO and Rh antigens. A compatibility chart is shown below. Plant and animal breeding. Over time, human beings have used genetic information to breed plant and animals by selectively choosing those with desired qualities. This process is called artificial selection. Through this process, plants and animals artificially breed in order to increase yields. The photographs show the high-yielding organisms. Improved resistance to pests and diseases. The photograph shows Ruiru 11 coffee breed, which is resistant to some diseases such as coffee berry disease. Increased rate of growth. Characteristics are selected and used to produce fast-maturing and high-yielding organisms. The photograph shows fast-growing and high-yielding maize varieties. Genetic counseling. This is the provision of information and advice on genetically inherited disorders, their risks and outcomes. The genetic counselor presents medical and scientific facts released so that those being counseled can make their own informed decision. Examples of disorders for which genetic counseling may be required include sickle cell anemia, albinism, hemophiliac, erythroblastosis fetalis, Klinefelter syndrome, and color blindness. The video shows a couple being counseled. Click on the play button to view the video. Genetic engineering. Genetic engineering refers to a method of changing the inherited characteristic of an organism in a predetermined by altering its genetic material. It involves identification of desirable genes, altering, isolating, and transferring it from one living organism to another. Genetic engineering has been applied in many areas including farming, medicine, environmental management, crime detection, and sequencing of genes. Farming. Genes for production of desirable characteristics have been isolated and transferred into plants to enable them process. These features include gene for synthesis of vitamin A has been transferred to rice in regions where rice is the only staple food. The photograph shows genetically modified rice. Genetically modified maize and soybeans have been produced which have resistance against insect pests. In maize production, wild types of maize have been engineered to produce a high-yielding and disease-resistant strains. The photograph shows genetically modified maize. Medicine. Human insulin has been produced from bacteria by transferring the gene for insulin production into bacteria. The genes for production of blood clotting factor has been transferred from man to embryo of a sheep to interact with the gene for milk production in order for the sheep to produce milk containing factor. This factor is later isolated and used to relieve hemophilia. Human somatotrophic hormone for treatment of dwarfism has now been extracted from genetically modified strain of E. coli bacteria. Production of vaccines. The vaccine for hepatitis B virus have been produced. The gene for the synthesis of the viral coat of hepatitis B is isolated and transferred to a fungus. The fungi produce this protein, which is later isolated and purified before injecting into man to stimulate production of antibodies against the virus. Gene therapy. Gene therapy is the replacement of faulty genes with the normal ones in order to correct the defects in man. Genetically modified organisms carry out the normal gene and introduce it into the affected tissue cells. For example, lungs cystic fibrosis is corrected by gene therapy. Environmental management. Decomposition of hydrocarbons in petroleum is used in controlling pollution due to oil spillage in water and soil by sheep through the use of pseudomonas. The photograph shows oil spillage. Crime detection. Particular individuals have specific DNA unique to them. Series of genetic techniques can be used to establish and produce a film on DNA patterns, producing a fingerprint. In a scene of crime, a specimen from a suspect, for example, hair, blood, semen, can be obtained and the DNA extracted and matched with suspects, helping isolate the culprit. DNA fingerprinting can also be used in solving disputed parentage since a child inherits one set of DNA from the mother and the other from the father. By comparing the DNA from the child, the mother and the suspect father can help identify the real father and confirm paternity. Cloning. This is a process of using an individual cell to produce a group of cells without fertilization. The offsprings are called clones. Clones are genetically identical. The illustration shows a picture of dolly sheep. Human genome. Genome refers to the total genetic content of any cell in an organism. Knowledge of genome is important in gene mapping. Gene mapping refers to identification of specific positions occupied by specific genes on a chromosome. Gene mapping can help in isolation and correction of defects such as sickle cell gene is located on chromosome 11. And hemophilia on the X chromosome. Sequencing of genes. The process involves analyzing the DNA to reveal the order of bases in all chromosomes, which assist in identification of defective genes, identification of genes that make individuals prone to certain diseases, alerting the individuals to make preventive measures, predict the amino acids, hence the proteins produced. This facilitates production of drugs to inhibit or enhance the process.